Alright, this is episode 2, and we've learned that blue and yellow sucks, basically. That's what we learned. But seriously, my name is, just so you know, this is RNA Lab, made by PBS. I guess that's our sponsor, PBS. And yeah, we'll be doing a playthrough of this, so... this is trial 1, I believe, and I couldn't even get past this. Okay. Now, if we do... Wait, why is this not working? Okay. It's, maybe it's not that bad. Maybe there's just some places that need it, and some places that don't. Guess that makes sense. Does that make sense? Oh! It's worked! Next puzzle. I don't remember why we need to do this. Oh, wait. Let's go back. If we go back home... Yes, I want to go back home. If we see the cell, this is explaining why we need to. The RNA snips off the tRNA's tail. Now the tRNA can collect an amino acid, take it to a protein-making machine, the ribosome. Next puzzle. Okay. RNA must fold into this shape. I'm not going in until you give me the shape. Alright. Let's go. Is this gonna work? That was easy. Wait, why didn't it help me at all? Like, I did it and nothing happened. Do I have to do it again? Wands can slide you the wrong way so when each side stack is one color. Switch between natural and target modes to see the slide. You can prevent this by swapping bonds. Try swapping the highlighted bond using the swap tool. RNA in target mode, click on the swap tool and then click on the bond. If you accidentally mispaint the bond, you can use the left arrow at the bottom of the screen to undo it. Swap. Any shape guide above turned white when you swap that base. Switch to natural mode to prevent that the stack is not sliding anymore. Congratulations, you fixed the slide. Next. Build part of our ribosome, RNA. So it's not rRNA, rRNA. R. And a bottle of rum. Okay, your RNA must fold into this shape. I'm not going to say the rest of that song. Try using only GC bonds for this puzzle. Only GC bonds. There's a story. Only GC bonds. Alright. This isn't very hard though, to be honest with you. Wait, what happened? So what if I use the swap tool? Does that fix the problem? Wait, no, this has... If I swap all of them, it should fix the problem, right? Nope. What if I red-green this bit? Nope, still not fix the problem. What if I swap this bit? Okay. Alright, I was right. Drag your RNA into the ribosome. So that's our other sponsor, the music producer. The rRNA has fixed part of the ribosome. Ribosome is made of two parts. You have to fix the small one, you make another rRNA to fix the large one. And I think this is the end. Yeah. 10 or more AU pairs. 10 or more AU pairs. GC strong. If you use too many, the RNA might bond to each other. Puzzle, use 10 or more AU bonds to help the RNA form. Will proper. Sometimes when you paint the base, the objective boxes above will not change, because they show what is happening in natural mode, not target mode. Even if you paint 10 AUs in target mode, this may not turn green into the RNA folds correctly. Alright. Let's go. Like, if I do this, is that gonna fix it? What if I do this? Is this gonna cause problems? No. Or this? No. How about this? Here? Or here? Well, that's good, but we need more AUs. Would this work? This? How about this? I don't know why we needed so many AUs, really. Drag your rRNA into the virtuous cell. We fixed it. Your rRNA has fully fixed the ribosome. All it needs now is a messenger RNA, MRNA, with the coded instructions. So what were they? Transfer, ribosome, and messenger. The coded instructions for building a protein. To finish the trial, you need to design an RNA that can help create mRNA. So you make the RNA, and I will make witty observations. Teamwork. Yeah, that's what teamwork is. That's what teamwork is, kids. Alright, build an mRNA splicer. Have 3 more UG pairs. These are the weakest bonds, but they are important. Present helpful in RNA, break its bonds and change shape. UG bonds work well in the middle stacks next to GC bonds. Work strategy will work for any puzzle that requires UG bonds. Turn all the RNA shape map white by painting the entire RNA molecule except for loops with GC bonds and using the swap tool to fix slides. Wrap the RNA shape map while place UGs one at a time. If the map ever turns red, undo that UG and try somewhere else. Okay. Can you shut up? If you're having trouble seeing the entire RNA, click and drag the background to move it around and use the magnifying glass at the bottom of the screen to zoom in and out. Sure, you're solving in target mode, not natural mode. Click and drag paint GC the entire RNA except for the loops. Don't move on until you get the green check mark. But you may need to swap some bonds to get it full right. UG bond to any hair middle of the stack, not at the end. The shape guide turns red, undo Z button, try again somewhere else. Otherwise, repeat until you have obtained 3 UGs. Try placing UGs at base 4, 1653, GCs everywhere else. Oh, I'm gonna write that down. That's sort of... Wait. Oh, no, I turned hints on. That's the problem. Well, why can't this guy shut up? How do I make this guy shut up? Well, I hate this. Why can't I do this? Like... How do I turn off the tutorials? Yes, I want to reset it. We're having problems. Forget it. I'm gonna reset it and make it so he's small. Because, frankly, I hate this person. Can I paint the bonds behind him is my question. No? Yes, indeed I can. So I can't make him shut up. I have to move myself. Frankly, you suck. Oh, I don't know what's going on. Why isn't that going? Do I have to swap this bit? How about this bit? No, maybe I do this and this. Okay. I'm gonna do this right now. This is good, but oh, there's one more bit I haven't actually done yet. I need to make this thing smaller so you guys can. Oh, zoom. There's a zoom. Yeah, you have to bash it when it freezes. I've learned. Alright, it's white, I think. Yep, it's white. Zoom out a lot. Okay, make it small, make it small. Has it turned red? Yay, I think I'm gonna be able to finish trial 1 today. No, you interrupt. Your mRNA splicing. Hello. Your mRNA splicing machine onto the nucleus. Yes! We've saved the cell. We've saved the cell. It is just one cell, but still. I like this. Congratulations, Grox. I seriously should have put CAD mode, shouldn't I? We have fixed protein synthesis in past trial 1. The mRNA splicer makes mRNA so the cell can make proteins. The mRNA leads the nucleus and travels to the ribosome where it reads the code message on the mRNA. And the ribosome turns the amino acids from the tRNAs into the polypeptide chain, folds into a protein. And now for something completely different. That my history teacher uses that phrase. And now for something completely different, RNA world. I'm just gonna hope that we meet this guy again and he produces important proteins for us. Part 1, complete achievement, protein synthesizer. Where does life come from, Grox? RNA brings us closer to ever than finding out. Than ever to finding out. Most biologists agree that RNA came before DNA and cells and made everything we think of as life possible. Starting in our virtual puddle, design 6 RNA molecules that will take you on a journey through several billion years of evolution from the early days of the RNA world all the way to the present. Now, brace yourself. Design an RNA that can copy itself. That's good. You can use to strengthen loops. We call these boosts. Simplest and most common boost is by placing a G at the end of a loop where it meets a stack. By placing a G where the arrow points. Alright, G coming up. G can only boost the loop if there's an A opposite to it on the other side. It's stacked with the G and A to form a weak bond in those positions. Boost the rest of the loops. Now bond the stack and turn the shape map white to solve the puzzle. Alright, let's do this. So this is the beginnings of life as we know it. So I can claim that I was the one who created life, because I did. Next, you drag yourself copying RNA into this primordial puddle. it's gonna create more of itself. It'll copy itself. This is awesome. RNA is using RNA building blocks, nucleotides in this virtual puddle to make copies of itself. So I made a cell, and now it's made I prepared a cell for the virtual lab, and now I'm going to create life on Earth. That's good. The RNA world theory suggests what? That billions of years ago, before cells existed, a self-copying RNA formed in a chaotic soup of molecules. But now we have a problem. The building blocks are running out. Quick, engineer an RNA that can build more nucleotides. The thing at stake is all life ever. No pressure. Wait, did we travel through time? I didn't realize. So RNA they can make RNA building blocks using nucleotides. Numbers next to RNA count pasted from top of the strand to the bottom. RNA folds up the numbers sitting next to the same bases. Booster work better when we place the G at the lowest number of nucleotides in a loop. So in the highlighted loop, place the G by the 5, not the 30. Place a G by the 5. Good, now boost the rest of the loops with G's at the lowest number of nucleotides and solve the rest puzzle by turning all the objective boxes green. What are objective boxes is my question. Okay. So which one's the lowest here? I think it's this. This here. And oh no. Oh no, I didn't mean to do that. I should turn this yellow again. Alright, so red off the places that should be red. Oh, what? So I think this is right. I think this is right. And then you go here and like here and then here and like here and then like here. Wait, why don't we have enough boost? 4 or more. I have 4 and more. I have 4 and I have even more. Alright, I'm confused. It's a good job I didn't design life on Earth today. So make sure you're solving in entirely mode, not natural mode. I know. Base is numbered 5. There's no 42. What are you even talking about? There's no 42. Okay, I'm not even gonna pretend to care. 66, 72, 100, 108, and 182. Green check mark. I've done this correctly. Paint GC bonds onto every pair on the stacks. Don't move until you get the green check mark on the shape guide. You may need to swap some bonds to get it to fold correctly. Alright, turn the hints back off again. And let's roll up the stacks. fix that bit. For the AU objective, AU bind into a pair in the middle of the stack. If the shape guide turns red, undo 0 and try again somewhere else. Repeat until you've painted 6 AU buttons. Right. You turn red. No reds. No reds. No reds. Alright. Painting 6. For the UG. UGs you pair in the middle stack. Turns red and try again. Otherwise defeat until you've painted 6 UGs. I don't think this is how things worked back then, but who cares? Let's just do it. Okay. I think I've saved life. Tag a nucleotide building RNA into a primordial puddle. Phew, just in time. Your nucleotide building tide building RNA fills the virtual puddle with new nucleotides. There, building blocks for creating more RNA machines. Millions of years passed, no big deal. It was now containing simple cells like this one. It has a membrane protects from the outside world, but it can't do much. Alright. I think we're signing off right about now. If you like this, please subscribe to my channel. And my name has been King Cadmos. And thanks for all the